In this video, we're going to talk about prokaryotic taxonomy or classification. Now, when talking about prokaryotic classification and identification, there's still a lot of discussions and changes still occurring. However, the authority on the subject is Bergey's manual. It was first developed in 1923 by David Hendricks Bergey, who is an American bacteriology. The printed version, or the hard copy, if you will, is on its 9th edition, but it has moved to an online edition as well. The one thing I would like to caution students is bacteria naming, bacteria classification is much more complex and is still in a state of development. So there are a lot of different thoughts on how to classify and name bacteria. In fact, it is that change that led from the major change in the Bergey's manual from their 8th to the 9th edition, so that it went from the manual of determinative bacteriology to the manual of systematic bacteriology. So just recognize that there are lots of different authorities on how to do this and thoughts and processes. So we're going to stick with Bergey's, and we're just going to learn high level of one way to classify. So one thing to point out, as I said, we have changed from being the determinative bacteriology to the systematic bacteriology, and why they did that was because it was what was the main information used for classification. When we were looking at it as a determinative bacteriology, we were using morphology, staining techniques, how bacteria reacted to various biochemical tests. So in other words, we were looking at the phenotypic traits. We're now moving toward the phylogenetic traits when classifying, identifying bacteria, and that means we're going to be looking at the genetics or the rRNA sequencing. However, we still do a lot with the phenotypic traits, and that helps us in classifying and identifying. And when you look at Bergey's, it will do those classifications based on like general shapes and morphology, gram staining, oxygen, motility, lots of different kinds of biochemical tests. This type of classification has led to basically prokaryotes being put into 4 major divisions: graciculites for the gram-negative, firmicutes for gram-positive, tourniquetes for those that don't have a cell wall, and mendoscutes for those that have a cell wall but don't use peptidoglycan. So since these terms are still used commonly today, let's talk just a little bit more about each. So graciculites, these are the gram-negative, and the gram-negative bacteria that fall into the graciculites are further divided into 4 groups based on their photosynthetic abilities. So if they're non-photosynthetic, they're called scotobacteria. If they're photosynthetic but do not produce oxygen, then they're anoxyphotobacteria. And if they are photosynthetic in the general way, meaning that they will produce oxygen in their photosynthetic processes, then they're considered oxophotobacteria. Now, gram-positive bacteria are called firmicutes, and they're divided into only 2 classes or 2 groups, and that's basically based upon their cell shape. So the Firmibacteria are those that are gram-positive: Coccus, Bacillus, the 2 shapes that you're used to, such as Streptococcus, Streptobacillus, etc. Whereas the Phthallobacteria are those that branch, or what we call filamentous bacteria. They might resemble mold. The other 2 groups, the tourniquetes, these are the bacteria that do not have cell walls at all. We discussed these previously in the cell wall video. These are going to be your mycoplasms, okay, and they fall in the group Mollicutes, and it's only one group there. The mendoscutes, on the other hand, do have cell walls, but they don't use peptidoglycan. And what you'll find with this group is these are mainly going to be those that are going to be the archaea bacteria, so in other words living in those extreme environments and can't have the typical cell wall structure because their environments will either break it down or it's not going to be formidable there. So those are the four main divisions based off of when we look at something from a phenotypic aspect. Berge's manual has, as I said, moved to more of a systematic bacteriology, and in this we're using that phylogenetic traits rather than the phenotypic traits as we did with starting with the first edition on up through the 8th. So the new Berge's has 5 volumes versus those 4. It's going to be looking more on ecological, okay, so what's going on with the ecology versus phenotype. It's also going to be looking at what does the group do basically clinically rather than pathogenic species being pulled out and being separated. So that is now where we are moving to. But again, bacteriology and classification is still in flux. It's a very fluid discussion amongst microbiologists, but we are moving more to the looking at genetics more so than the morphology.